Hey student, here we are again to learn about human reproduction and pregnancy events. You will get separate video about zygote to gestation and parturition. See that too. Do like and subscribe. Summary of human reproductive events. Gametogenesis. In males, production of sperms, spermatogenesis, occurs in the testes. In females, production of ova, oogenesis, occurs in the ovaries. Insemination, transfer of sperms into the female reproductive tract during sexual intercourse. Fertilization, fusion of the male and female gametes, usually in the fallopian tube, forming a zygote. Blastocyst formation and implantation. The zygote divides to form a blastocyst, which implants in the uterine wall. Gestation, pregnancy, embryonic and fetal development inside the uterus over approximately 9 months. Parturition, childbirth, the process of delivering the baby. Notable differences between male and female reproductive roles. Males. Spermatogenesis begins at puberty and can continue into old age. Reproductive role is primarily limited to sperm production and insemination. Females. Oogenesis begins before birth, with ovum maturation resuming at puberty and ceasing around menopause, 50 years. Females undergo pregnancy and childbirth, involving significantly more physiological changes. 4 main parts of the male reproductive system, with ducts: testes, scrotum, accessory ducts, reedy testis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, urethra, penis. Male accessory glands, support reproduction. Pair of seminal vesicles. Prostate gland. Pair of bulbourethral glands, Cooper's glands. Testes. Detailed description. Location and position. The testes, singular testis, are paired male gonads located outside the abdominal cavity in a pouch called the scrotum. The scrotum acts as a thermoregulatory organ, maintaining a temperature about 2 to 2.5 degrees Celsius lower than the normal body temperature, which is essential for spermatogenesis, sperm production. Shape and size. Each testis is oval in shape. Length, 4 to 5 centimeters. Width, 2 to 3 centimeters. Covering. The testes are enclosed in a dense fibrous capsule called the tunica albuginea. This protective covering extends inward to form septa, dividing each testis into about 250 testicular lobules. Testicular lobules. Each lobule contains 1 to 3 highly coiled seminiferous tubules, which are the functional units of sperm production. Seminiferous tubules. These are lined by 2 main types of cells. Male germ cells, spermatogonia. These are diploid cells located at the basal layer. They undergo meiotic divisions to produce haploid sperms. Sertoli cells provide nutritional and structural support to developing sperm cells, also called nurse cells, help in the phagocytosis of residual cytoplasm during sperm maturation. Interstitial Leydig cells, found in the interstitial spaces between seminiferous tubules. These cells secrete androgens, mainly testosterone, regulate the development of secondary sexual characters and maintenance of male reproductive organs, are endocrine in nature. The interstitial spaces also contain blood capillaries, lymphatic vessels, immunologically active cells, accessory ducts, reedy testis, vasa efferentia, carry sperm from reedy testis to epididymis. Epididymis, located on posterior testis, stores sperm. Vas deferens, ascends into abdomen, loops over bladder. Ejaculatory duct, formed by union with duct from seminal vesicle, opens into urethra. Urethra, runs through penis, ends at urethral meatus, opening. Function, ducts store and transport sperm from testis to outside. Functions of the testes. Spermatogenesis, formation of mature sperm cells. Endocrine function, secretion of male sex hormones, androgens, primarily testosterone. Developmental role, Testosterone stimulates the development of male reproductive organs and secondary sexual characteristics, like deep voice, facial hair, muscle mass. Male reproductive glands. Detailed explanation. There are 3 types of male accessory glands. Gland number, location, main function. Seminal vesicles 2 paired. Behind the bladder produce 60% of semen volume. Prostate gland 1 single. Below the bladder adds enzymes, citric acid. Bulbourethral, Cooper's. Glands 2 paired. Below prostate. Near base of penis secretes mucus for lubrication. 1. Seminal vesicles. Number paired 2. Location. Posterior side of urinary bladder. Duct. Each seminal vesicle has a duct that joins the vas deferens to form the ejaculatory duct. Secretion. Thick alkaline viscous fluid. Contains fructose, prostaglandins, fibrinogen. Functions. Fructose. Energy source for sperms. Prostaglandins. Stimulate uterine contractions to aid sperm movement. Alkaline pH. Neutralizes acidic environment of female vagina. Makes up about 60 to 70% of semen volume. 2. Prostate gland. Number single. Location. Below urinary bladder. Surrounds upper part of urethra. Structure. Chestnut-shaped gland made of alveoli and ducts. Secretion. Milky, slightly acidic fluid. Contains citric acid, calcium, enzymes, like prostate-specific antigen PSA. Functions. Activates sperm motility. Helps liquefy semen after ejaculation, fibrinolysin. Forms about 25 to 30% of semen volume. 3. Bulbourethral, Cooper's, glands. Number paired 2. Location. At the base of the penis, beneath the prostate. Ducts. Open into the urethra. Secretion. Clear, slippery mucus, pre-ejaculate. Functions. Lubricates urethra for smooth sperm passage. Neutralizes acidic traces of urine in urethra. Secreted before ejaculation. Overall contribution to semen. Seminal plasma. Seminal vesicles 60 to 70%. Prostate gland 25 to 30%. Bulbourethral glands. Small volume but crucial for lubrication. Semen equals sperm plus seminal plasma. Semen provides a nourishing, protective, and transport medium for spermatozoa. Summary table. Gland secretion main components function. Seminal vesicle alkaline fluid fructose. Prostaglandins nourish sperm, aid transport. Prostate gland milky fluid citric acid. PSA activates sperm, liquefies semen. Bulbourethral mucus mucoproteins lubrication. Neutralizes urine. Structure of penis. External male copulatory organ. Made up of erectile tissue, spongy tissues with blood spaces. Contains the urethra, common passage for semen and urine. Parts. Root, attached to the pelvic wall. Body, shaft. Cylindrical part. Glans penis. Enlarged tip, highly sensitive. Prepuce, foreskin. Fold of skin covering the glands. Functions of penis. Copulation. Delivers sperm into the female reproductive tract during sexual intercourse. Urination. Excretes urine from the body through urethra. Erection. Erectile tissue fills with blood during sexual arousal, enabling penetration. Semen ejaculation. Passage of semen through urethra during climax, orgasm. Structure of the penis. Male external genital organ. Glans penis. Enlarged, rounded tip of the penis. Highly sensitive, aids in sexual stimulation. Covered by a loose fold of skin called foreskin or prepuce. Prepuce foreskin. A retractable fold of skin covering the glans. Can be removed in circumcision. Shaft. Body of penis. Cylindrical part made of erectile tissues. Contains 3 columns of erectile tissue. 2 corpora cavernosa, dorsal side. 1 corpus spongiosum, ventral side surrounds urethra. Urethra. Runs through the penis. Inside corpus spongiosum. Common passage for urine and semen. Root of penis. The base of the penis attached to the pubic bone. Anchors penis to body. Erectile tissue. Spongy tissue that fills with blood during sexual arousal. Causes erection required for intercourse. The penis is a unique organ in the male reproductive system, specially designed for two key functions: urination and sexual reproduction. Let's explore the different tissues that make up its structure. 1. Erectile tissues. The main functional core. Most of the penis is made of soft, spongy tissue known as erectile tissue. This tissue is filled with blood vessels and acts like a sponge. It absorbs blood and becomes rigid during an erection. 2. Corpora cavernosa. These are paired cylindrical columns on the upper side. They're the largest and are mainly responsible for the stiffness of the penis during erection. 1. Corpus spongiosum. This lies below, along the urethra. It also fills with blood, but stays slightly softer. This protects the urethra and allows semen or urine to pass through easily. It also forms the glans, the rounded tip of the penis. 2. Tunica albuginea. The protective covering. Surrounding the erectile tissues is a strong, elastic layer called the tunica albuginea. It acts like a sheath that supports the penis during erection, maintains its shape, prevents overexpansion. 3. Skin and subcutaneous tissue. The outer shield. The penis is covered by a thin, soft, and stretchable skin. At the tip, this skin forms a fold over the glands known as the foreskin or prepuce. This layer is rich in sensory nerve endings, making the penis highly sensitive to touch. 4. Blood vessels and nerves. The power supply. During arousal, blood enters through arteries and fills the erectile tissues, causing erection. Veins then help drain the blood afterward. Nerves play a crucial role too. They carry signals of sensation and trigger erection and ejaculation responses. In summary, the penis is a marvel of design. Its erectile tissues help with rigidity, connective tissues offer strength and structure, and the skin and nerves add sensitivity and protection, making it perfectly suited for its dual role in reproduction and urination. For more such human biology explanations, don't forget to like, share, and subscribe to the channel. 2.2 The female reproductive system. Detailed explanation. The female reproductive system is designed to produce ovum, support fertilization, provide an environment for embryo development, enable childbirth, support child care through mammary glands. Main components: organ function. Ovaries produce ovum and ovarian hormones. Oviducts, fallopian tubes, transport ovum, site of fertilization. Uterus site for implantation and fetal development. Cervix connects uterus to vagina, passage for sperm and baby. Vagina birth canal and copulatory organ. External genitalia protects reproductive tract. Includes vulva. Mammary glands produce milk for infant care, functional part of reproductive system. Ovaries, the primary sex organs. Number paired. Location: one on each side of the lower abdomen. Size: 2 to 4 centimeters in length. Attachment: connected to the uterus and pelvic wall via ligaments. Functions: produce female gametes, ova. Secrete steroid hormones. Estrogen and progesterone. Structure of ovary: covered by thin germinal epithelium. Internal tissue, stroma, is divided into cortex, outer region, contains developing ovarian follicles in various stages. Medulla, inner region, contains blood vessels, lymphatic vessels, and nerve fibers. In today's session, we'll understand the female reproductive system, focusing on the accessory ducts and external genitalia. Let's begin. 1. Female accessory ducts. These ducts help in transporting gametes, facilitating fertilization, and supporting the embryo. The key components are oviducts, also known as fallopian tubes. Each oviduct is about 10 to 12 centimeters long. They connect the ovary to the uterus and are the site of fertilization. Let's break down the parts of the oviduct. Infundibulum. This is the funnel-shaped part closest to the ovary. Catches the ovum after ovulation. Fimbriae. These are finger-like projections extending from the infundibulum. They help sweep the released ovum into the oviduct. Ampulla. The central, widest region of the oviduct. This is where fertilization usually occurs. Isthmus, a narrow, muscular part that connects to the uterus. It pushes the zygote toward the uterus for implantation. Uterus, the womb. It is a single, pear-shaped organ located in the pelvic cavity. Supported by ligaments, it is the site of implantation and embryo development. The wall of the uterus has 3 layers. Perimetrium, outer thin membrane. Acts as a protective layer. Myometrium, thick smooth muscle layer. Contracts strongly during childbirth. Endometrium, inner glandular lining. Thickens each month. Sheds during menstruation if no pregnancy occurs. Cervix and cervical canal. Cervix is the narrow lower part of the uterus that opens into the vagina. Cervical canal connects the uterus and vagina. It allows passage of sperm during intercourse. Menstrual flow. Delivery of the baby during childbirth. Vagina. This is a muscular elastic canal. Functions. Receives the penis during sexual intercourse. Serves as a passageway for menstrual flow. Along with the cervical canal, forms the birth canal. Let's now explore the female external genitalia, also called the vulva. This includes visible structures at the opening of the female reproductive tract. Each part plays an important role in protection and function. 1. Mons pubis. Right above the pubic bone is a soft, fatty cushion called the mons pubis. It's covered with hair after puberty and provides a protective layer for the delicate organs underneath. 2. Labia majora. These are the outer, thick, fleshy folds of skin that surround the vaginal opening. They act like protective flaps, keeping the inner structure safe from infection, injury, or friction. 3. Labia minora. Tucked just under the labia majora are the labia minora, smaller inner folds of skin. These protect both the urethral opening and the vaginal orifice, helping maintain hygiene and shield from external particles. 4. Hymen. Covering part of the vaginal opening in some females is a thin membrane called the hymen. While traditionally linked to virginity, it's not a reliable indicator. It can break due to various non-sexual activities like sports or falls. 5. Clitoris. At the top junction where the labia minora meet is a tiny structure called the clitoris. It's rich in nerve endings, making it highly sensitive, and plays a major role in sexual arousal and pleasure. In summary, the vulva isn't just about reproduction. It's a protective gate, a sensory organ, and an essential part of female anatomy. Each structure, from the mons pubis to the clitoris, works together for comfort, hygiene, and function. Important notes about the hymen. Can tear due to: first, sexual intercourse, physical activities, cycling, horse riding, sports, tampon use or accidental injury. Presence or absence of hymen is not a reliable sign of virginity. Fallopian tubes, oviducts. Detailed explanation for Neet. Introduction. Fallopian tubes, also known as oviducts or uterine tubes, are paired muscular tubes in the female reproductive system. Length: 10-12 cm. Function: transport ovum egg from ovary to uterus. Site for fertilization of the egg by the sperm. Structure of a fallopian tube. Each fallopian tube has 3 main regions, from ovary to uterus. Region description function. Infundibulum funnel-shaped. Open end near the ovary captures ovum after ovulation. Fimbriae finger-like projections at the edge of infundibulum sweep ovum into the fallopian tube. Ampulla broad, middle portion site of fertilization. Sperm meets ovum. Isthmus narrow part joining uterus transports fertilized egg, zygote to uterus. Histological layers of fallopian tube. Each fallopian tube wall is made up of 3 layers. Layer description. Mucosa, inner. Lined with ciliated columnar epithelium that helps move the ovum. Muscularis, middle. Smooth muscle, circular and longitudinal, that contracts to aid ovum/zygote movement. Serosa, outer. Thin protective membrane covering the tube. Functions of the fallopian tube. Collection of ovum from the ovary using fimbriae. Site of fertilization, usually in the ampulla region. Transport of ovum or zygote to the uterus using ciliary movement. Muscular contractions. Provides nourishing secretions to support egg/sperm/zygote temporarily. Clinical relevance. Condition description. Ectopic pregnancy, fertilized egg implants in fallopian tube. Dangerous. Tubal blockage leads to infertility. Egg or sperm can't travel. Tubectomy, surgical cutting of fallopian tubes as a permanent birth control method. Here are the detailed descriptions of the 3 major clinical conditions associated with the fallopian tubes. Ectopic pregnancy, tubal blockage, and tubectomy, suitable for NEET/board exam. 1. Ectopic pregnancy. Definition. Ectopic pregnancy is a medical condition in which the fertilized egg, zygote, implants outside the uterus, most commonly in the fallopian tube, also called a tubal pregnancy. Why it happens. Due to blockage, scarring, or inflammation in the fallopian tube, the zygote fails to reach the uterus. It gets implanted within the tube itself, which is not structurally capable of supporting fetal growth. Consequences. The growing embryo can rupture the tube, causing internal bleeding, severe pain, and can be life-threatening. Immediate surgical or medical treatment is needed. Symptoms. Sharp abdominal pain, vaginal bleeding, dizziness or fainting due to blood loss. Treatment. Surgical removal of the embryo or damaged tube. Medication like methotrexate if caught early. 2. Tubal blockage. Fallopian tube obstruction. Definition. A condition where one or both fallopian tubes are blocked, preventing the egg and sperm from meeting. Causes. Pelvic inflammatory disease, PID, due to bacterial infection. Endometriosis, growth of uterine lining outside the uterus. Scar tissue from previous surgeries or ectopic pregnancies. Tuberculosis, genital TB in some regions. Effect on fertility. If both tubes are blocked, natural pregnancy is not possible. If one tube is blocked, chances of conception are reduced. Diagnosis. HSG, hysterosalpingography. An X-ray test with contrast dye. Sonohysterography. Laparoscopy. Treatment options. Tuboplasty, surgical removal of blockage. IVF, in vitro fertilization, if blockage is severe. 3. Tubectomy, female sterilization. Definition. Tubectomy is a surgical procedure in which fallopian tubes are cut and tied/sealed, preventing the egg from reaching the uterus. Purpose. Permanent birth control, contraception, in women. Part of family planning programs in many countries. Procedure. Can be done via laparoscopy, keyhole surgery, or mini-laparotomy. The fallopian tubes are cut, tied, or sealed to block the passage of ova. Advantages. Highly effective, over 99%. No long-term hormonal side effects. Disadvantages. Irreversible. Though reversal surgeries exist, success is not guaranteed. Minor surgical risks, bleeding, infection. Who should opt? Women who do not want more children. Should be chosen with full awareness and consent. Summary table. Condition description, consequences, treatment/management. Ectopic pregnancy, implantation of zygote in fallopian tube, life-threatening. Internal bleeding, surgery or methotrexate. Tubal blockage, fallopian tubes blocked, one or both. Infertility or reduced fertility, tuboplasty, IVF. Tubectomy, surgical sterilization, cutting tubes. Permanent contraception, laparoscopic surgery. Mammary glands. Detailed explanation. Introduction. Mammary glands are the functional and characteristic feature of all female mammals. In humans, they are present as paired structures called breasts, located over the pectoral muscles of the chest. They become fully functional after childbirth, producing milk to nourish the newborn, a process called lactation. Structure of the mammary glands. Each breast contains 1. Glandular tissue, the core component responsible for milk production, divided into 15 to 20 lobes per breast. 2. Mammary lobes. Each lobe is made up of many alveoli clusters. Alveoli are the actual milk-secreting cells, surrounded by myoepithelial cells that contract to push milk out. 3. Alveoli. Microscopic sac-like structures where milk is secreted and stored temporarily in their lumens, cavities, rich in secretory epithelial cells. 4. Mammary tubules. Alveoli of each lobe drain into small tubules. These tubules combine to form larger mammary ducts. 5. Mammary ducts. Collect and transport milk from the lobes. Several mammary ducts converge to form a mammary ampulla. 6. Mammary ampulla. A widened storage chamber just before the nipple. Temporarily stores milk before release. 7. Lactiferous duct. Connects the ampulla to the nipple. Milk is sucked out through these ducts during breastfeeding. Histological composition. Component description. Alveolar cells secrete milk into lumens. Myoepithelial cells contract to push milk from alveoli to ducts. Ducts lined by epithelial tissue to aid transport. Fatty tissue surrounds glandular tissue, giving breast its shape and size. Connective tissue supports and binds the lobes. Hormonal regulation of mammary glands. Hormone role. Estrogen and progesterone promote development of breast tissue during puberty and pregnancy. Prolactin from pituitary stimulates milk production after childbirth. Oxytocin from pituitary triggers contraction of myoepithelial cells, milk ejection, letdown reflex. Functions of the mammary glands. Milk production, lactation. To feed the newborn. Passive immunity. Through antibodies in colostrum, first milk. Mother-infant bonding. Physical and emotional connection during breastfeeding. Regulates infant nutrition. Complete balanced diet for the first 6 months. Clinical relevance. Condition description. Mastitis infection/inflammation of breast tissue, often during lactation. Breast cancer abnormal growth of cells in mammary tissue. Galactorrhea excessive or inappropriate milk production. Inverted nipple may hinder breastfeeding in some cases. Summary chart. Part function. Alveoli milk secretion and storage. Mammary tubules transport milk from alveoli. Mammary ducts merge tubules to central duct. Mammary ampulla stores milk before release. Lactiferous duct delivers milk to nipple. 2.3 Gametogenesis. Detailed explanation. What is gametogenesis? Gametogenesis is the biological process by which gametes, sex cells, are formed in sexually reproducing organisms. In males, this process is called spermatogenesis, formation of sperms. In females, it's known as oogenesis, formation of ova or eggs. Spermatogenesis in males. Location. Occurs in the seminiferous tubules of the testes. Begins at puberty under the influence of hormones. Sequence of events in spermatogenesis. Spermatogonia 2N. Diploid male germ cells located at the inner wall of seminiferous tubules. Undergo mitotic divisions to increase their number. Primary spermatocyte 2N. Some spermatogonia enlarge and differentiate. These cells undergo meiosis I, reduction division. Secondary spermatocytes N. 2 haploid cells formed from one primary spermatocyte. Undergo meiosis II. Spermatids N. 4 haploid cells from meiosis II. Small non-motile immature spermatozoa sperms. Formed from spermatids by spermiogenesis, transformation process. Mature motile gametes capable of fertilization. Final stage. Spermiation. The release of mature sperms from Sertoli cells into the lumen of seminiferous tubules. Hormonal control of spermatogenesis. Hormone source and function. GnRH secreted from hypothalamus. GnRH stimulates pituitary to release FSH and LH. FSH is secreted from anterior pituitary that acts on Sertoli cells which supports spermiogenesis. LH is secreted by anterior pituitary, acts on Leydig cells and initiates secretion of androgens, testosterone. Testosterone secreted from Leydig cells, which stimulates spermatogenesis process. Let's now explore the structure of a human sperm. The male gamete responsible for fertilizing the ovum. A sperm is a microscopic, highly specialized motile cell, and is divided into 4 distinct parts, each with a specific function. 1. Head. The head is the front portion of the sperm. It contains an elongated haploid nucleus, which carries the father's genetic material, 23 chromosomes. Covering the head is the acrosome, a cap-like structure filled with powerful enzymes such as hyaluronidase and acrosin. These enzymes help the sperm penetrate the egg's protective layers during fertilization. 2. Neck. The neck is a small region that connects the head to the middle piece. It contains the centriole, which plays a vital role after fertilization. It helps form the mitotic spindle that supports the first divisions of the zygote. 3. Middle piece. This part is packed with mitochondria. These mitochondria generate ATP, energy, required for movement. This energy is used to power the tail and help the sperm swim through the female reproductive tract. 4. Tail. The tail is a long flagellum, making the sperm motile. Produces whip-like movements. This allows the sperm to propel forward, helping it reach and fertilize the ovum. Sperm is a motile, microscopic, flagellated male gamete. Important data. Each ejaculate 200 to 300 million sperms. For fertility, greater than or equal to 60% should have normal shape and size. Greater than or equal to 40% should show vigorous motility. Semen composition. Component description. Sperm's male gametes, produced in testes. Seminal plasma secretions from seminal vesicle, prostate, and bulbourethral glands. Functions: nourish, protect, and transport sperm. Support maturation and motility. Key terms. Term meaning. Spermatogenesis, formation of sperms from spermatogonia. Spermiogenesis, transformation of spermatids to spermatozoa. Spermiation, release of mature sperms from Sertoli cells. Androgens, male sex hormones, mainly testosterone. Sertoli cells provide nutrients and support for developing sperms. Oogenesis. Formation of female gamete, ovum. Definition. Oogenesis is the biological process of formation and development of a mature female gamete or ovum in the ovaries. Unlike spermatogenesis, oogenesis begins before a female is born, pauses at different stages, and is completed only after fertilization. Let's see stages of oogenesis. Stage, event, time of occurrence. Phase 1. Multiplication phase. Millions of oogonia, which are diploid 2N cells, are formed by mitosis from germinal epithelium. This occurs during fetal life, even before girl is born. Phase 2. Growth phase. Diploid oogonia, the mother cells, grow and enter prophase, one of meiosis cell division, and known as primary oocytes, but remains arrested, occurs in fetal life before birth. Phase 3. Arrested phase. Primary oocytes remain arrested in prophase 1 till puberty, occurs from birth to puberty. Phase 4. Maturation phase. Every menstrual cycle, one primary oocyte resumes meiosis 1 and forms one secondary haploid oocyte, N, and first polar body, N, occurs at puberty. Phase 5. Ovulation. Secondary oocyte is released from ovary within Graafian follicle. Occurs at mid of menstrual cycle. Phase 6. Meiosis 2. Completed only if fertilization occurs, forming ovum, N, and second polar body, N, after fertilization. Follicle development. Follicle type description. Primary follicle, primary oocyte surrounded by a single layer of granulosa cells. Secondary follicle, more layers of granulosa cells. Appearance of theca layer. Tertiary follicle has fluid-filled cavity, antrum. Theca differentiates into theca interna and theca externa. Graphian follicle, fully mature follicle containing secondary oocyte with zona pellucida. Ovulation, Graphian follicle ruptures to release secondary oocyte. Important cellular products. Primary oocyte, 2N arrested in prophase I. Secondary oocyte, enlarged cytoplasm-rich cell, due to unequal division. Polar bodies. Small, cytoplasm poor, non-functional, may degenerate. Why unequal division? To ensure that the secondary oocyte retains most of the cytoplasm, rich in nutrients, essential for early embryonic development. Let's compare oogenesis and spermatogenesis. 1. When does it begin? Oogenesis begins before birth. Yes, you heard that right. A female baby is already carrying all her primary oocytes even before she's born. Spermatogenesis, on the other hand, starts at puberty in males and continues throughout life. 2. Arrest phases. In oogenesis, the primary oocyte gets arrested in prophase I before birth. Later, meiosis II is paused and only completed after fertilization. In contrast, spermatogenesis is a continuous process. No arrest phases once it starts at puberty. 3. Number of gametes produced. Oogenesis is conservative. Out of one primary oocyte, it gives rise to one ovum and 2 or 3 non-functional polar bodies. Spermatogenesis, however, is productive. It produces 4 functional sperms from each primary spermatocyte. For cytokinesis, in oogenesis, the cell division is highly unequal. The ovum receives almost all the cytoplasm, and the polar bodies are left with minimal content. In spermatogenesis, cytokinesis is equal, distributing cytoplasm uniformly among the spermatids. 5. Cytoplasm allocation. Ovum gets maximum cytoplasm to support early development after fertilization. In spermatogenesis, cytoplasm is shared equally, because each sperm is small, mobile, and doesn't need stored nutrients. 6. When is the process completed? Oogenesis is completed only after fertilization. Spermatogenesis completes before ejaculation, so the sperms are already ready for fertilization. Hormonal control of oogenesis. Hormone and role. 1. GnRH secreted from hypothalamus. Stimulates LH and FSH secretion. 2. FSH stimulates follicle growth. 3. Hormone, LH, triggers ovulation and Graphian follicle maturation. 4. Estrogen and progesterone secreted by follicles and corpus luteum regulate cycle and uterus lining. 2.3. Oogenesis and follicular development. Part 1. Oogenesis. Formation of female gamete, ovum. Oogenesis is the process of formation of a mature ovum from oogonia inside the ovary. It starts during fetal development and completes only after fertilization. Stages of oogenesis. Stage cell type ploidy event. 1. Multiplication phase oogonia diploid, 2N. Formed by mitotic division from primordial germ cells during fetal life. 2. Growth phase primary oocyte diploid, 2N. Grows in size, stores nutrients, enters meiosis I and is arrested at prophase I. 3. Maturation phase, at puberty. Secondary oocyte plus first polar body haploid. N. Primary oocyte resumes meiosis I during each menstrual cycle. Meiosis I is unequal. 4. Ovulation secondary oocyte haploid, N. Released from ovary at day 14 of menstrual cycle. 5. Fertilization if occurs. Ovum plus second polar body haploid, N. Meiosis II completes only after sperm entry. Polar bodies. Formed due to unequal division of cytoplasm. Do not develop into ova. May degenerate or remain. Purpose of unequal division. to ensure that the ovum retains maximum cytoplasm and nutrients for early embryonic development. Part 2. Follicular development, folliculogenesis, occurs alongside oogenesis. Each developing ovum is enclosed in a follicle, which also undergoes structural changes. Stages of follicle development. Follicle type contains features. Primordial follicle contains primary oocyte. It is single flat layer of granulosa cells. Primary follicle contains primary oocyte. It is enlarged oocyte plus single layer of cuboidal granulosa cells. Secondary follicle contains primary oocyte, multiple granulosa layers plus development of theca layer. Tertiary follicle contains enlarged primary oocyte. Here formation of antrum, fluid-filled cavity, occurs plus theca layer divided into theca interna and theca externa. Graphian follicle contains secondary oocyte. This is mature follicle ready for ovulation. Oocyte surrounded by zona pellucida and corona radiata. Hormonal control. Hormone source function. GnRH hypothalamus stimulates FSH and LH from pituitary. FSH pituitary promotes follicle development. LH pituitary triggers ovulation. Converts follicle to corpus luteum. Estrogen follicles prepares endometrium. Progesterone corpus luteum maintains pregnancy if fertilization occurs. Oogenesis is the production line for the female gamete, the ovum. It kicks off before birth, when a female is still in the womb. The starring cells here are the oogonia, which transform into primary oocytes, waiting patiently until puberty. From then on, each menstrual cycle gives a few oocytes a chance to mature. The grand finale. Ovum formation. Folliculogenesis, on the other hand, is all about building the perfect home for that maturing oocyte, the follicle. It, too, begins before birth, but picks up its pace monthly from puberty to menopause. It's driven by granulosa and theca cells, creating a nurturing environment. The climax. Ovulation, when the follicle bursts to release the oocyte. And what's pulling the strings behind the scenes? The same hormonal trio. GnRH, FSH, and LH, orchestrating both acts in perfect hormonal harmony. Oogenesis is the biological process by which the female gamete, or ovum, is formed inside the ovaries. It is a complex process that begins even before the female is born and is only completed upon fertilization. Oogenesis starts during fetal development when millions of gamete mother cells, called oogonia, are formed within the developing ovaries. These oogonia are diploid and multiply by mitosis. Soon after their formation, they begin meiosis but stop at the first stage of meiosis 1, prophase I. At this arrested stage, they are now called primary oocytes. Each primary oocyte becomes surrounded by a single layer of flat follicular, granulosa, cells to form the primordial follicle. From birth to puberty, a large number of these follicles degenerate in a process called atresia, leaving only around 60,000 to 80,000 primary follicles in each ovary by the time of puberty. At puberty, under the influence of follicle-stimulating hormone, FSH, some of these primordial follicles grow into primary follicles. The granulosa cells become cuboidal and proliferate, and the oocyte also grows in size. These follicles then develop into secondary follicles, in which multiple layers of granulosa cells appear and a specialized structure called the theca layer begins to form. As development continues, the follicle becomes a tertiary follicle, characterized by the formation of a fluid-filled cavity called the antrum. The theca layer organizes into two layers: the theca interna, which secretes hormones, and the theca externa, protective outer layer. During this stage, the primary oocyte completes the first meiotic division, resulting in the formation of a large secondary oocyte and a tiny first polar body. This division is highly unequal, with the secondary oocyte retaining most of the nutrient-rich cytoplasm. The secondary oocyte now becomes surrounded by a new membrane called the zona pellucida. The tertiary follicle matures into the Graafian follicle, which eventually ruptures and releases the secondary oocyte from the ovary. This process is known as ovulation, which occurs around the 14th day of a typical menstrual cycle. The secondary oocyte starts the second meiotic division but halts at metaphase 2. It will complete this division only if fertilization occurs. If a sperm penetrates the oocyte, meiosis II completes, producing a mature ovum and a second polar body. The ovum is now ready to fuse with the sperm nucleus to form a zygote. While the oocyte undergoes these changes, the surrounding follicular cells also undergo follicular development, which occurs alongside oogenesis. Each stage of follicle development provides nourishment, hormonal support, and a suitable environment for the developing oocyte. The maturation of follicles and the oocyte is regulated by hormones such as gonadotropin-releasing hormone, GnRH, from the hypothalamus, which stimulates the anterior pituitary to secrete FSH and luteinizing hormone, H. FSH primarily promotes follicle growth, while LH triggers ovulation and the formation of the corpus luteum from the ruptured Graafian follicle. The corpus luteum secretes progesterone, which prepares the uterus for possible implantation and pregnancy. If fertilization does not occur, the corpus luteum degenerates, hormone levels fall, and menstruation begins. In summary, oogenesis is a prolonged and cyclical process that begins before birth and concludes with ovulation and fertilization, while follicular development supports and nourishes the maturing oocyte through each stage until ovulation. Follicular development stages. Primordial follicle. Primary oocyte plus single flat granulosa cell layer. Primary follicle. Primary oocyte grows. Granulosa cells become cuboidal and multiply. Secondary follicle. More layers of granulosa cells appear. Theca layer begins to develop outside granulosa cells. Tertiary follicle. Appearance of fluid-filled antrum. Theca differentiates into theca interna, secretes hormones, estrogens. Theca externa, protective function. Graafian follicle, fully mature follicle, contains secondary oocytes surrounded by zona pellucida, ready for ovulation. Menstrual cycle, overview. Definition. The reproductive cycle in female primates, e.g., monkeys, apes, humans, is known as the menstrual cycle. Menarche. The first menstruation at puberty is called menarche. Marks the beginning of the reproductive phase. Cycle duration. Typically lasts 28 to 29 days. It is a monthly cycle from the start of one menstruation to the next. Ovulation timing. One ovum is released during mid-cycle, around 14th day. Phases of the menstrual cycle. 1. Menstrual phase, days 1 to 5. Menstrual bleeding occurs, caused by shedding of the endometrial lining and rupture of blood vessels. Occurs only if fertilization has not taken place. Average duration 3 to 5 days. 2. Follicular proliferative phase, days 6 to 13. Primary follicles grow into a mature Graafian follicle in the ovary. Endometrium regenerates and thickens, stimulated by FSH and LH from the pituitary gland. Estrogens secreted by growing follicles. LH and FSH levels gradually rise, peaking around day 14. 3. Ovulation day 14. LH surge leads to rupture of the Graafian follicle. Ovum is released into the fallopian tube. Marks the transition to the luteal phase. 4. Luteal secretory phase, days 15 to 28. Remaining Graafian follicle converts into corpus luteum. Corpus luteum secretes progesterone, helps maintain a thick, vascular endometrium, prepares uterus for implantation if fertilization occurs. If no fertilization, corpus luteum degenerates, progesterone levels fall, endometrium breaks down, leading to next menstruation. Menstruation stops when fertilization occurs. Menstrual cycle is arrested during pregnancy. Endometrium is maintained for embryo implantation. Menopause. Around 50 years of age, menstrual cycles cease permanently. Known as menopause. Marks the end of the reproductive phase. Key points. Menarche menopause equals reproductive age. Cyclic menstruation indicates a normal, healthy reproductive system. Lack of menstruation may indicate pregnancy, or may result from stress, illness, or malnutrition. 2.4 Menstrual cycle. NCERT Class 12 Biology Notes. Definition. Term description. Menstrual cycle described as the cyclic reproductive process in female primates, involving ovulation and menstruation. Next, term menarche, described as the first occurrence of menstruation at puberty. Next term is menopause, described as the permanent cessation of menstruation around 50 years of age. And last term is cycle duration, described as approximately 28 to 29 days. Varies slightly among individuals. Let's break down the 4 phases of the menstrual cycle, a beautifully coordinated monthly process preparing the female body for pregnancy. 1. Menstrual phase. Day 1 to 5. This is when menstruation occurs. The endometrial lining of the uterus is shed, leading to bleeding through the vagina. This happens due to a sharp decrease in estrogen and progesterone, as the corpus luteum degenerates. 2. Follicular phase, proliferative phase. Day 6 to 13. Now, the body begins to prepare again. Primary follicles in the ovary begin to grow and mature into a Graafian follicle. Meanwhile, the endometrial lining regenerates. This is due to a rise in FSH and LH, and increasing estrogen levels from the developing follicles. 3. Ovulation phase. Day 14. On day 14, the mature Graafian follicle ruptures and releases the ovum. This is known as ovulation. It is triggered by a sharp spike in LH levels, known as the LH surge. 4. Luteal phase, secretory phase. Day 15 to 28. After ovulation, the empty follicle converts into the corpus luteum. This structure secretes progesterone, which thickens the endometrium in preparation for implantation. FSH and LH levels drop now, while progesterone rises. In summary, the menstrual cycle is controlled by hormones and includes menstruation, follicular growth, ovulation, and preparation for pregnancy. If fertilization doesn't occur, the cycle starts again. Let's understand how the menstrual cycle is controlled by a beautifully synchronized hormonal system. These hormones act like messengers to guide each step of the cycle. 1. GnRH, gonadotropin-releasing hormone. Source: hypothalamus. Function: it acts as the master signal. GnRH stimulates the anterior pituitary to release FSH and LH, kickstarting the menstrual cycle. 2. FSH, follicle-stimulating hormone. Source: anterior pituitary. Function: FSH helps grow ovarian follicles, one of which becomes the dominant Graafian follicle that will release the ovum. 3. Luteinizing hormone. Source: anterior pituitary. Function: LH causes the rupture of the Graafian follicle, leading to ovulation. It also helps form the corpus luteum, the structure that secretes progesterone. 4. Estrogen. Source: developing ovarian follicles. Function: estrogen helps rebuild the endometrium after menstruation and also supports follicular development in the ovary. 5. Progesterone. Source: corpus luteum. Function: progesterone stabilizes and thickens the endometrial lining, making it ready to receive a fertilized egg for implantation. In summary, this hormonal interplay, GnRH, FSH, LH, estrogen, and progesterone, works like a symphony, orchestrating each phase of the menstrual cycle and preparing the body for possible pregnancy. Event after ovulation. Scenario outcome. Fertilization occurs, corpus luteum persists. Progesterone maintains endometrium, no menstruation. No fertilization, corpus luteum degenerates. Drop in progesterone, endometrial breakdown, menstruation begins again. Important terms to remember. Term description: Graafian follicle, mature ovarian follicle ready to release an ovum. Corpus luteum, hormone-secreting structure formed from the ruptured follicle after ovulation. Endometrium, inner lining of the uterus. Supports embryo if implantation occurs. Surge sudden rise in LH level around day 14, triggering ovulation. Summary. Menstrual cycle occurs in all female primates. 4 key phases. Menstrual, follicular, ovulation, luteal. Regulated by a complex interplay of hormones. GnRH, FSH, LH, estrogen, progesterone. Cyclic menstruation continues from menarche to menopause. Absence of menstruation may indicate pregnancy or medical condition. 2.5 Fertilisation and implantation. Class 12 Biology Notes. 1. Fertilization. Definition and overview. Term description. Fertilization. The fusion of a sperm with an ovum to form a diploid zygote. Next term, site of fertilization. Ampullary region of the fallopian tube. Next term, insemination. Release of semen into the vagina during coitus. Next term, zygote. Diploid cell formed after fertilization. Contains 46 chromosomes. 2 steps in fertilization process. Step description. 1. Insemination. Semen containing sperm is ejaculated into the vagina. 2. Sperm movement. Motile sperms swim through cervix, uterus, ampullary region of the fallopian tube. 3. Ovum transport. Released ovum also reaches ampullary region. Fertilization can occur only if sperm and ovum arrive simultaneously. 4. Sperm-ovum interaction. One sperm binds with zona pellucida of the ovum and penetrates it. 5. Polyspermy prevention. Changes in ovum membrane prevent entry of additional sperms. 6. Acrosomal reaction. Acrosomal enzymes help sperm penetrate ovum membrane. 7. Completion of meiosis II. Secondary oocyte completes meiosis II, forms haploid ovum plus second polar body. 8. Fusion of nuclei. Haploid nuclei of sperm and ovum fuse, diploid zygote is formed. Have you ever wondered who determines whether a baby will be a boy or a girl? 3. Sex determination in humans. Humans have 46 chromosomes, 44 autosomes and 2 sex chromosomes. In females, the sex chromosome pair is XX. In males, it's XY. During fertilization, the ovum always carries an X chromosome. The sperm, however, can carry either an X or a Y chromosome. So what happens when sperm type combines with ovum X? Resulting baby. X chromosome X plus X XX female baby. Y chromosome X plus YXY male baby. Conclusion. The father determines the sex of the baby, because only he can contribute either an X or a Y chromosome through his sperm. So blaming the mother for the baby's gender is scientifically incorrect. Remember, gender determination is purely a biological process, not a matter of choice or blame. Let's spread awareness, not myths. For cleavage and embryo formation. After the fusion of sperm and ovum, a magical process begins. The zygote undergoes cleavage, a rapid series of mitotic divisions. These divisions produce blastomeres, the small daughter cells. After a few rounds we get a morula, a solid ball of 8 to 16 cells. The morula continues to divide and hollows out to form a blastocyst. The blastocyst has 2 key parts. Trophoblast, the outer layer, helps the embryo attach to the uterus. Inner cell mass. These cells will form the actual embryo. 5. Implantation process. The journey continues as the blastocyst reaches the uterus. Now what? Attachment. The trophoblast sticks to the thick uterine wall, endometrium. Cell proliferation. Uterine cells start rapidly dividing, forming a protective cover over the blastocyst. Embedding. The blastocyst burrows into the endometrium. Result. Implantation is complete, and pregnancy officially begins. Summary points to remember. Fertilization occurs in the ampullary region of the fallopian tube. Only one sperm fertilizes the ovum, thanks to the zona pellucida barrier. The resulting zygote is diploid and marks the start of a new individual. Sex is decided at fertilization. X equals girl. Y equals boy. Embryonic development. Cleavage, morula, blastocyst, implantation. Once implantation is successful, the uterus nurtures the growing baby for the next 9 months. 2.5 Fertilization and implantation. Mixed format notes. What is fertilization? Fertilization is the fusion of a haploid sperm and a haploid ovum to form a diploid zygote. It occurs in the ampullary region of the fallopian tube. For fertilization to occur, both the ovum and sperms must meet simultaneously in the ampullary region. Process of fertilization, step by step. Insemination. During copulation, semen is ejaculated into the female vagina. Sperm transport. Sperms move through the cervix, uterus, ampulla of the fallopian tube. Ovum transport. Ovum released from the ovary also travels to the ampulla via fallopian tube. Sperm-ovum contact. Sperm binds to the zona pellucida of ovum and triggers chemical changes to block polyspermy, entry of more than one sperm. Penetration. Acrosomal enzymes help the sperm penetrate ovum's membranes. Meiotic completion. Secondary oocyte, arrested in metaphase 2, now completes meiosis, forms haploid ovum plus second polar body. Nuclear fusion. Male and female nuclei fuse, forms diploid zygote, 2n equals 46. Concept highlight. Sex determination. Gametes chromosome type outcome. Ovum always X. Sperm X or Y. X, sperm, plus X ovum, XX female baby. Y sperm, plus X ovum, XY male baby. Hence, father determines the sex of the baby, not the mother. Cleavage and early development. Zygote undergoes cleavage, rapid mitotic divisions. Forms 2, 4, 8, 16 cell stages. These cells are called blastomeres. Morula. Solid ball of 8 to 16 blastomeres. Blastocyst. Hollow structure formed from morula with 2 parts. Trophoblast. Outer layer helps in implantation. Inner cell mass. Develops into the embryo. Summary of stages after fertilization. Stage description. Cleavage mitotic divisions of zygote. Forms blastomeres. Morula 8 to 16 cell stage. Solid ball of cells. Blastocyst fluid-filled structure with trophoblast and inner cell mass. Implantation attachment and embedding of blastocyst in endometrium. What is implantation? Trophoblast attaches to the endometrial lining of uterus. Uterine cells divide rapidly and cover the blastocyst. The blastocyst embeds itself in the endometrium. This marks the beginning of pregnancy. Key points to remember. Fertilization occurs in the ampulla of fallopian tube. Acrosome reaction and zona pellucida prevent multiple sperm entry. Meiosis II in oocyte is completed only after sperm entry. Formation of diploid zygote initiates life. Sex of the child is determined at the time of fertilization. Implantation is the embedding of the blastocyst into the uterus lining. 2.6 Pregnancy and embryonic development. Placenta formation and function. What happens after implantation? After the blastocyst implants in the endometrium, finger-like projections called chorionic villi develop from the trophoblast. These villi become surrounded by uterine tissue and maternal blood. Formation of placenta. Chorionic villi plus uterine tissues equals placenta, structural plus functional unit. Placenta facilitates exchange of nutrients, gases, and waste. Protection of embryo. Hormonal support for pregnancy. The umbilical cord connects the embryo to the placenta and acts as a lifeline between mother and fetus. Functions of placenta. Function type description. Respiratory supplies O2 and removes CO2. Nutritive transports glucose, amino acids, lipids, vitamins, minerals to fetus. Excretory removes nitrogenous waste, urea, ammonia. Endocrine produces hormones, HCG, HPL, estrogen, progesterone, relaxin. Immunological provides passive immunity to fetus. Some antibodies cross. 2 hormones in pregnancy. Major hormones produced. Human chorionic gonadotropin HCG. Maintains corpus luteum. Basis of pregnancy test. Human placental lactogen HPL. Stimulates milk gland development and fetal growth. Estrogen and progesterone. Support endometrium, suppress menstrual cycle, promote uterine growth. Relaxin. Secreted by ovary in later stages. Relaxes pelvic ligaments for delivery. Other hormones with increased levels in pregnancy. Hormone function. Cortisol supports fetal organ maturation and metabolism. Prolactin prepares mammary glands for milk production. Thyroxine maintains metabolic rate and supports fetal brain development. 3. Embryonic development. Month-wise features. Month major events. End of first month heart develops and starts beating, detectable via stethoscope. End of second month formation of limbs and digits. Arms, legs, fingers. By third month, 12 weeks, most major organs formed. Genitalia visible, first trimester ends. By fifth month, first fetal movements felt by mother. Hair appears on scalp. By sixth month, 24 weeks. Eyelids separate, eyelashes form, body covered in fine hair. By ninth month, fetus fully developed and ready for delivery. 4. Germ layer formation and organ development. Formation of 3 germ layers. After implantation, the inner cell mass differentiates into ectoderm, skin, brain, spinal cord, eyes. Endoderm, gut lining, liver, lungs. Mesoderm, muscles, bones, heart, blood, kidneys. These layers give rise to all tissues and organs in the adult body. Stem cells. Inner cell mass contains stem cells. These are pluripotent. Can give rise to any tissue or organ in the body. Summary chart. Human pregnancy timeline. Trimester weeks, key developmental events. First 0 to 12 heartbeat, limbs, genitalia, major organs. Second 13 to 24 movement, hair growth, facial features. Third 25 to 40 rapid growth, organ maturity, delivery. Key points to remember. Placenta is vital for fetal nutrition, respiration, excretion, and hormone production. HCG confirms pregnancy in tests. First heartbeat is detectable at end of first month. Movement felt around fifth month. Stem cells from inner cell mass are pluripotent. Pregnancy lasts 9 months, 280 days. In humans. 2.7 Parturition and lactation. 1. Parturition, childbirth. Definition. Parturition is the process of expelling the fully developed fetus, childbirth, from the uterus at the end of pregnancy. The average duration of human pregnancy is 9 months, also called gestation period. Neuroendocrine reflex in parturition. How is parturition triggered? Signals for parturition arise from fully developed fetus, placenta. These signals initiate mild uterine contractions called fetal ejection reflex. This reflex stimulates the maternal pituitary to release oxytocin, a hormone. Oxytocin acts on the uterine muscles, causes stronger uterine contractions. These contractions stimulate further oxytocin release. This is a positive feedback loop. Uterine contractions, oxytocin, more contractions, more oxytocin. Result: expulsion of the baby through the birth canal, followed by placenta expulsion. Table. Hormonal mechanism of parturition. Event: hormone involved effect. Fetal ejection reflex initiates parturition signal. Maternal response: oxytocin induces strong uterine contractions. Positive feedback loop: oxytocin contractions, oxytocin more contractions. Delivery of baby ends uterine contractions. Placenta expelled completes parturition. Doctors may inject synthetic oxytocin to medically induce labor. Lactation. What is lactation? Lactation is the production and release of milk from the mammary glands to feed the newborn baby. Initiated towards the end of pregnancy. Continues after childbirth. Role of mammary glands. Undergo differentiation during pregnancy. Begin milk production at the end of gestation. Colostrum. The first milk. Feature description. Name colostrum. Timing first few days post-delivery. Content high levels of antibodies, proteins, and nutrients. Importance boosts immunity in the newborn. Essential for protection. Lactation. Hormonal control. Hormones secreted by function. Prolactin pituitary gland stimulates milk production. Oxytocin pituitary gland stimulates milk ejection. Letdown reflex. Breastfeeding is highly recommended in early infancy for strong immunity, healthy growth, mother-child bonding. 3. Summary flowchart. Fetal maturity, fetal ejection reflex, maternal pituitary, oxytocin. Uterine contractions, oxytocin, stronger uterine contractions. Expulsion of baby, parturition. Placenta expelled, mammary glands stimulated, lactation starts. Colostrum secretion, infant gains immunity. Key points to remember. Parturition is triggered by neuroendocrine signals and involves a positive feedback loop of oxytocin and uterine contractions. Lactation ensures nutrition and immunity to the baby. Begins with colostrum secretion. Hormones involved. Oxytocin, for both childbirth and milk ejection. Prolactin, for milk production. Here are the answers and solutions for the exercises from NCERT Class 12 Biology Chapter 2. Human reproduction. 1. Fill in the blanks. A. Humans reproduce sexually. B. Humans are viviparous. C. Fertilization is internal in humans. D. Male and female gametes are haploid. E. Zygote is diploid. F. The process of release of ovum from a mature follicle is called ovulation. G. Ovulation is induced by a hormone called LH, luteinizing hormone. H. The fusion of male and female gametes is called fertilization. 1. Fertilization takes place in ampullary region of fallopian tube. J. Zygote divides to form blastocyst which is implanted in uterus. K. The structure which provides vascular connection between fetus and uterus is called placenta. 4. Two major functions each of testis and ovary. Testis. First, the testis performs two crucial roles. Spermatogenesis. The production of sperm. Hormone secretion. Mainly androgens like testosterone which maintain male sexual characteristics. Ovary. Similarly, the ovary plays two major roles in females. Oogenesis. The formation of the ovum or egg. Secretion of hormones like estrogen and progesterone, which regulate the menstrual cycle and support pregnancy. 5. Structure of a seminiferous tubule. Inside the testis are coiled structures called seminiferous tubules, the site of sperm production. Lined by germinal epithelium, each tubule contains spermatogonia, stem cells that begin spermatogenesis, primary and secondary spermatocytes, spermatids, and spermatozoa at different stages. Sertoli cells, which provide nourishment and support. Outside the tubule, interstitial cells or Leydig cells secrete testosterone, essential for this process. 6. Spermatogenesis. Step-by-step process. Spermatogenesis is the process of forming haploid sperm from diploid spermatogonia. Happens in 4 steps. Spermatogonia divide by mitosis to form primary spermatocytes. These undergo meiosis I to form secondary spermatocytes. Secondary spermatocytes undergo meiosis II to form spermatids. Spermatids then undergo a transformation called spermiogenesis to become mature spermatozoa. 7. Hormones involved in spermatogenesis. This complex process is hormonally regulated by the following. FSH, follicle-stimulating hormone, stimulates Sertoli cells and spermatogenesis. H, luteinizing hormone, stimulates Leydig cells to secrete testosterone. Testosterone directly supports the development of sperm and secondary sex traits. Spermiogenesis and spermiation. Spermiogenesis is the transformation of non-motile spermatids into fully developed, motile spermatozoa with head, midpiece, and tail. Spermiation is the final release of these mature sperm from the Sertoli cells into the lumen of the seminiferous tubule, where they are ready to travel through the male reproductive tract. 10. Major components of seminal plasma. Seminal plasma is the fluid part of semen, rich in nutrients and substances essential for sperm survival. It contains fructose, provides energy for sperm movement, secreted by seminal vesicles. Prostaglandins, aid in sperm motility and movement through the female tract. Citric acid and enzymes, from the prostate, help maintain sperm viability. Mucus, from bulbourethral glands, acts as a lubricant during ejaculation. These secretions mix with sperm to form semen, a powerful medium for fertilization. 11. Functions of male accessory ducts and glands. Now, what are the roles of ducts and glands in male reproduction? Accessory ducts like the epididymis, vas deferens, and ejaculatory duct store, nourish, and transport sperm from testes to urethra. Accessory glands include seminal vesicles, provide fructose-rich secretion. Prostate gland adds enzymes and buffers. Bulbourethral glands secrete mucus for lubrication. Together, they ensure sperm are protected, nourished, and mobile for successful fertilization. 12. What is oogenesis? A brief account. Now let's shift focus to the female gamete, the ovum. Oogenesis is the process of egg formation in females. Before birth, germ cells in the ovary multiply to form oogonia. These become primary oocytes and are arrested in prophase I of meiosis. Puberty. Each menstrual cycle, one primary oocyte resumes meiosis and completes meiosis I. It forms a secondary oocyte and a first polar body, with little cytoplasm. At ovulation, the secondary oocyte is released. Meiosis II completes only if fertilization occurs, resulting in an ovum and second polar body. So, oogenesis begins before birth, pauses, resumes at puberty, and finishes only if fertilization happens. 15. Functions. Corpus luteum secretes progesterone to maintain endometrium. B. Endometrium, site for implantation, supports embryo. C. Acrosome, contains enzymes to penetrate ovum. D. Sperm tail, helps in motility. E. Fimbriae, help capture the ovum from the ovary. 16. True/false, correct the false. False. Androgens are produced by Leydig cells. B. True. Sertoli cells support and nourish spermatids. C. False. Leydig cells are found in testis, not ovary. E. True. Leydig cells produce testosterone. E. False. Oogenesis occurs in the ovary, not corpus luteum. F. True. The menstrual cycle stops during pregnancy. G. True. The hymen can break due to many reasons, not necessarily intercourse. 17. What is menstrual cycle? Regulating hormones. The menstrual cycle is the monthly reproductive cycle, 28 days, in women. It's regulated by FSH, LH, estrogen, and progesterone, with GnRH controlling FSH and LH release. 18. What is parturition? Hormones involved. Parturition is childbirth. It's triggered by oxytocin released in response to uterine contractions, fetal ejection reflex. The positive feedback loop between oxytocin and uterine contractions leads to delivery. 19. Why blaming women for daughter is wrong. In humans, mothers always produce eggs with an X chromosome, but fathers produce sperm with either X or Y. The sex of the baby is determined by which sperm fertilizes the egg, fathers. So yes, it's scientifically wrong to blame women. 20 eggs released in a month. Humans usually release 1 egg per cycle. In identical twins, that 1 egg splits after fertilization, so 1 egg released. For fraternal twins, 2 eggs are released and fertilized separately. 21 eggs released by a dog with 6 puppies. Dogs are polyticous, meaning they release multiple eggs. To have 6 puppies, the ovary would have released at least 6 eggs, possibly more. Some MCQ for revision you can think answer. 1. Humans reproduce asexually. B. Sexually. He both. None. 2. Humans are classified as oviparous. B. Viviparous. C. Ovoviviparous. D. Hermaphrodites. 3. Fertilization in humans is external. B. Internal. C. Both internal and external. D. Self-fertilization. 4. Male and female gametes are diploid. B. Haploid. C. Triploid. D. Polyploid. 5. A zygote is haploid. B. Diploid. C. Triploid. D. Tetraploid. 6. The release of an ovum from a follicle is called implantation. B. Spermiogenesis. C. Ovulation. D. Fertilization. 7. Ovulation is induced by B. C. Estrogen. D. Progesterone. The fusion of gametes is known as cleavage. B. Fertilization. C. Implantation. D. Spermiation. 9. Fertilization occurs in ovary. B. Uterus. C. Ampullary region of the fallopian tube. D. Cervix. 10. The zygote divides to form implants in the uterus. Blastomere. B. Morula. C. Blastocyst. D. Gastrula. 11. The vascular connection between fetus and uterus is called amnion. B. chorion. T. placenta. D. umbilical cord. 12. Sperm production occurs in epididymis. B. Seminal vesicles. C. Seminiferous tubules. D. Vas deferens. 13. Leydig cells secrete estrogen. B. Testosterone. C. Progesterone. D. Oxytocin. 14. Sertoli cells are important for producing testosterone. B. Nourishing developing sperm. C. Producing estrogen. D. Making the acrosome. 15. The acrosomal cap on sperm contains DNA. B. Enzymes for penetration. T. Mitochondria. Ribosomes. 16. Semen contains secretions from which gland for lubrication? Prostate. B. Seminal vesicle. C. Bulbourethral gland. D. Pituitary. 17. The first polar body forms during which process? Spermatogenesis. B. Oogenesis. C. Cleavage. D. Implantation. 18. Which hormone maintains the endometrium during the luteal phase? B. C. Estrogen. D. Progesterone. 19. The LH surge triggers menstruation. B. Follicle formation. C. Ovulation. D. Fertilization. 20. The embryo implants in the myometrium. B. Perimetrium. C. Endometrium. D. Fallopian tube. 21. The functional unit of the placenta is the blastocyst. B. Chorionic villus. C. lumen. Yolk sac. 22. During parturition, oxytocin is released to stimulate A. Milk production. B. Uterine contractions. T. Follicle growth. D. Fertilisation. 23. The initial milk, rich in antibodies, is called colostrum. B. Lactose. T. Skim milk. Whey. 24. Which hormone is primarily responsible for milk production? Oxytocin. B. Prolactin. C. Estrogen. D. FSH. 25. The fetus begins showing visible movement by month. Third. B. Fourth. T. Fifth. D. Sixth. 26. The outer layer of the blastocyst that forms the placenta is A. Inner cell mass. B. Epiblast. C. Trophoblast. D. Blastocele. 27. Chorionic villi are part of embryo only. B. Uterine tissue. T. Placenta. D. Endometrium only. 28. Which structure connects the placenta to the embryo? Amnion. B. Yolk sac. C. Umbilical cord. D. chorion. 29. Which hormone is not secreted by the placenta? A. Human chorionic gonadotropin, HCG. B. Human placental lactogen, HPL. C. Estrogen. D. Follicle-stimulating hormone, FSH. 30. Which hormone is released later in pregnancy from the ovary? Estrogen. B. Progesterone. C. Relaxin. D. HPL. 31. The hormone that supports breast development and milk production is prolactin. B. Oxytocin. 32. The 3 germ layers in the embryo are formed from blastomere. B. Inner cell mass. C. Trophoblast. D. Epiblast. 33. The ectoderm gives rise to muscles. B. Nervous system. C. Digestive tract. D. Blood vessels. 34. The mesoderm gives rise to skin. B. Neural tissue. C. Skeletal system. D. Respiratory tract lining. 35. Stem cells from the inner cell mass are totipotent. B. Pluripotent. C. Unipotent. D. Differentiated. 36. The fetal heartbeat can be first heard after 1 week. B. 1 month. E. 2 months. E. 3 months. 37. Formation of limbs and digits occurs by the end of first month. B. Second month. C. Third month. D. Fifth month. 38. External genitalia become distinct by 6th week. B. 9th week. C. 12th week. D. 20th week. 39. Fine body hair and eyelash formation occurs during first trimester. B. Second trimester. C. Third trimester. D. At birth. 40. What initiates the process of parturition? Mother's pituitary signals. B. Fully developed fetus and placenta. C. Ovarian follicles. D. Amniotic sac rupture. 41. The hormone directly responsible for uterine contractions is oxytocin. B. Prolactin. C. Estrogen. D. Relaxin. 42. Which reflex enhances oxytocin release during parturition? Sucking reflex. B. Ejection reflex. C. Fetal ejection reflex. D. Dilation reflex. 43. Colostrum is rich in lipids. B. Carbohydrates. C. Antibodies. D. Hormones. 44. Breastfeeding is recommended after birth because it's a tradition. B. It contains antibodies. C. It reduces pregnancy chances. D. It's cheaper than formula. 45. Colostrum is produced before pregnancy. B. During ovulation. C. During initial days of lactation. D. After menopause. 46. What maintains pregnancy by preventing uterine contractions early on? Oxytocin. B. HCG. C. Progesterone. D. 47. Which hormone maintains the corpus luteum in early pregnancy? H. B. HCG. C. Estrogen. D. Relaxin. 48. Which hormone is released in large amounts just before labor? Estrogen. B. Progesterone. C. Oxytocin. D. Prolactin. 49. The process of milk ejection is controlled by prolactin. B. Estrogen. C. Oxytocin. D. Cortisol. 50. The hormone that inhibits further ovulation during pregnancy. Estrogen. B. HCG. C. Progesterone. D. HOTS questions. Human reproduction, Class 12 Biology. 1. Question: why does a sharp increase in LH level trigger ovulation instead of FSH, even though both are involved in follicular development? Answer: LH surge leads to weakening of the follicular wall and stimulates the rupture of the Graafian follicle, causing ovulation. FSH supports follicular growth but does not induce rupture. Question: a woman has blocked fallopian tubes. How will it affect fertilization and embryo development? Answer: fertilization typically occurs in the ampullary region of the fallopian tube. A blockage prevents the meeting of sperm and ovum, thus hindering fertilization. No zygote or embryo will form. 3. Question: despite the sperm and egg both being haploid, why is only the maternal mitochondria inherited by the zygote? Answer: the mitochondria in the sperm tail do not enter the egg during fertilization. Only the egg contributes cytoplasm and organelles, including mitochondria, to the zygote, leading to maternal inheritance. 4. Question: explain how hormonal imbalance in a female could affect the menstrual cycle and fertility. Answer: imbalance in FSH, LH, estrogen or progesterone can disrupt follicular development, ovulation, endometrial thickening, and corpus luteum maintenance. This leads to irregular cycles and infertility. 5. Question: why is colostrum considered a natural vaccine for newborns? Answer: colostrum is rich in IgA antibodies that protect the newborn against infections by enhancing passive immunity. Question: a couple is unable to conceive. Investigations reveal that the male partner has low testosterone. How will this affect spermatogenesis? Answer: testosterone, secreted by Leydig cells under LH stimulation, is essential for spermatogenesis. Low testosterone results in decreased sperm production and male infertility. 7. Question: why do doctors recommend avoiding pregnancy until at least 6 weeks after delivery? Answer: after childbirth, hormonal levels are unstable and the uterus needs time to return to normal size. Also, ovulation and the menstrual cycle may resume unpredictably. Physical recovery and lactation also play roles. Question: how does the feedback mechanism of oxytocin during labor differ from most hormonal feedback systems? Answer: oxytocin during labor is regulated by a positive feedback loop, where uterine contractions stimulate more oxytocin release, intensifying contractions until birth. Most hormones work on negative feedback. Question: 2 babies are born from the same pregnancy. 1 placenta is observed. What type of twins are they likely to be? Answer: identical, monozygotic, twins, as they originate from one zygote and usually share a placenta. Question: why is a zygote called totipotent, but inner cell mass is pluripotent? Answer: the zygote can give rise to all cell types including extraembryonic tissues like placenta. Thus, it is totipotent. Inner cell mass gives rise to all body tissues but not extraembryonic tissues, hence pluripotent. Here are assertion and reason type questions. AR 4 Class 12 Biology. Chapter 2. Human Reproduction, designed to build higher-order thinking and conceptual clarity. Brain assertion and reason questions, with answers and explanations. 1. Assertion: A. Ovulation is triggered by a sudden surge in luteinizing hormone, LH. Reason: LH stimulates the conversion of the ruptured follicle into corpus luteum. Answer: A and R are true, and R is the correct explanation of A. Explanation: LH surge causes rupture of the Graafian follicle, ovulation, and the same LH helps the remaining follicular tissue form the corpus luteum. Assertion: A fertilization in humans is internal. Reason: Our sperms are motile and can swim to meet the ovum in the uterus. Answer: A is true, R is false. Explanation: fertilization occurs in the ampullary region of the fallopian tube, not in the uterus. Assertion: a human zygote is diploid. Reason: our zygote is formed by fusion of two diploid gametes. Answer: A is true, R is false. Explanation: the zygote is diploid 2n, but gametes are haploid n. Assertion: A, only the mother's mitochondria are inherited by the offspring. Reason: R, the sperm contributes mitochondria during fertilization. Answer: A is true, R is false. Explanation: although sperm contains mitochondria in the tail, only the nucleus enters the egg, hence only maternal mitochondria are inherited. Assertion: a colostrum is beneficial for newborns. Reason: R, it is rich in IgA antibodies which provide passive immunity. Answer: A and R are true, and R is the correct explanation of A. Explanation: colostrum helps fight infections in newborns due to the presence of immunoglobulins, especially IgA. Assertion: a spermatogenesis is initiated at puberty. Reason: our testosterone levels rise significantly during puberty. Answer: A and R are true, and R is the correct explanation of A. Explanation: increased secretion of GnRH, LH, and FSH leads to rise in testosterone, initiating spermatogenesis. Assertion: A, the placenta produces hormones like HCG and HPL. Reason: R, these hormones are essential for maintaining pregnancy. Answer: A and R are true, and R is the correct explanation of A. Explanation: HCG maintains corpus luteum, HPL promotes fetal growth and metabolism. Assertion: oogenesis is completed before a female is born. Reason: R, all primary oocytes are formed during fetal life and remain arrested in prophase I. Answer: A is false, R is true. Explanation: oogenesis starts before birth but is completed after fertilization. Primary oocytes are arrested in prophase I at birth. Assertion: a spermiogenesis and spermiation are different processes. Reason: R, spermiogenesis is the process of releasing mature sperm from Sertoli cells. Answer: A is true, R is false. Explanation: spermiogenesis is the transformation of spermatids into spermatozoa. Spermiation is the release of sperm from Sertoli cells. Assertion: a female reproductive events are cyclic. Reason: our menstrual cycle regulates release of ovum, preparation of uterus, and hormonal balance. Answer: A and R are true, and R is the correct explanation of A. Explanation: hormonal feedback between the ovary and uterus regulates the menstrual cycle every month. Long answer questions. Human reproduction. 1. Describe the structure and functions of the human male reproductive system. Points to cover. Diagram of male reproductive system, labeled. Testes. Structure: seminiferous tubules, Leydig cells, and functions. Male accessory ducts. Rete testis, vasa efferentia. Epididymis, vas deferens. Male accessory glands. Seminal vesicles, prostate, bulbourethral glands. Penis. Structure and function. Function of seminal plasma. 2. Explain the process of spermatogenesis. Add a labeled diagram showing different stages. Points to cover. Definition: formation of sperm from germ cells in testes. Location: seminiferous tubules. Phases: multiplication, mitosis of spermatogonia. Growth: primary spermatocyte formation. Maturation: meiosis I and tube spermatids. Spermiogenesis: spermatids, spermatozoa. Permeation: role of hormones. GnRH, FSH, LH, testosterone. Neat labeled diagram showing stages. 3. Describe the menstrual cycle in human females. Explain the roles of hormones involved. Points to cover. Definition: cyclic changes in female reproductive system. 28-day cycle. Phases. Menstrual phase, day 1 to 5. Follicular/proliferative phase, day 6 to 13. Ovulatory phase, day 14. Luteal/secretory phase, day 15 to 28. Hormones involved: FSH, LH, estrogen, progesterone. Corpus luteum and feedback regulation. Diagram. Graph of hormone levels. 4. Describe the process of fertilization and implantation in humans. Points to cover. Fertilization. Definition and site. Ampullary-isthmic junction. Events. Insemination. Capacitation. Acrosomal reaction. Fusion of gametes. Zygote formation. Zygote morula blastocyst. Implantation. Process and timing. Role of trophoblast and uterine endometrium. 5. Describe the structure and functions of placenta. Mention the hormones it secretes. Points to cover. Definition and development. Chorionic villi plus uterine tissue. Functions. Exchange of gases, nutrients, and wastes. Protection. Hormone secretion. Hormones. CG. FL. Estrogens. Progesterone. Relaxin. Umbilical cord function. 6. Explain the process of parturition and the role of hormones in it. Points to cover. Definition. Expulsion of fully developed fetus. Neuroendocrine reflex mechanism. Role of fetal ejection reflex. Hormones. Oxytocin, from maternal pituitary uterine contractions. Positive feedback loop of oxytocin. Expulsion of baby and placenta. 7. What is oogenesis? Describe the stages of oogenesis in detail. Points to cover. Definition. Formation of ovum in ovaries. Prenatal stage. Formation of primary oocytes. Postnatal stage. Arrest at prophase I until puberty. Meiosis I. Secondary oocyte and first polar body. Meiosis II. Ovum and second polar body after fertilization. Role of hormones. FSH and LH. Diagram showing stages. And that brings us to the end of this chapter on human reproduction. From gamete formation to fertilization, implantation, and birth. You've just explored one of the most fascinating journeys of life. Remember, every concept you learn here is not just part of your syllabus, but part of understanding life itself. If you found this video helpful, make sure to like, share, and subscribe to stay updated with all upcoming chapters and science concepts made simple. Got questions? Drop them in the comments. I'm here to help you learn better. Until next time, keep learning, stay curious, and believe in the power of knowledge.